This is Selma Schimmel at the 14th World Conference on Lung Cancer, WCLC, which is organized by the International Association of the Study of Lung Cancer, the IASLC, in Amsterdam, the Netherlands. Our discussion begins with Dr. Marileila Varella Garcia. Welcome. Dr. Garcia is Professor of Medical Oncology in the Department of Medicine, the University of Colorado Anschutz Medical Campus in Aurora, Colorado. Hello, Dr. Garcia.
Nice to be here.
What percentage of cancer patients, non-small cell lung cancer patients, have this mutation?
It's about 5% of non-small cell lung cancer. But the non-small cell lung cancer has two big subgroups, what we call adenocarcinoma and what we call squamous cell carcinoma. If we only take adenocarcinoma, is about 10%.
Which is more, so that's more common.
Yeah. The adenocarcinoma is 60% of the non-small cell lung cancer.
So a patient, before they get treated today, has to get tested.
Yes, because there are specific inhibitors for this protein, the ALK protein, as well as there are specific inhibitors for a number of other proteins already. But patients whose tumor depends on ALK, is driven by ALK. Right. When they take this inhibitor is pretty specific. So what the inhibitor does is that destroy that specific protein, nothing else. So only destroy then the cells that are damaging, that have the mechanism of gene expression without control. Only the cells that are growing driven by ALK is going to be affected by the ALK inhibitors. And then the tumor shrinks. When we learn that ALK inhibitor was shrinking lung cancer advanced stage, was unheard of, and shrinks the tumor to 80%, sometimes to 90%.
To test for the mutation, is it blood or tissue that the doctor works with?
It's tissue. It's tissue because the abnormality only happens in the lung tumor tissue, in the lung tissue that turned to be tumor.
And the way in which the pathologist does the analysis would be through a biopsy?
It can be a resection if the patient was surgically resectable. Mm-hmm. It can be a biopsy, any type of biopsy. Core needle biopsy, aspirates, does not matter for the testing. And it's possible that we also get pleural effusion, for instance. So there are multiple biological sources of tumor cells that can be used for the test.
They can take aspirate some of the liquid. Exactly. And do it.
Exactly. And then if the pleural effusion contains tumor cells,
yes,
we can find the abnormality in that cells.
So what does this mean for our understanding of lung cancer in general for the future? So now the ALK pathway and this mutation has been identified, which is really changing the way in which patients get treated, because now it's not just arbitrary treatment, there's a methodology involved. Where's research going in the identification of other pathways that could be influential in lung cancer?
We have been focusing multiple pathways, but before we get there, when we develop any test and we discover this, so this test was completely discovered less than 3 years ago. And the first process is to do standardization, because we don't want to be the only institution or a few institutions only are able or qualified to do the test.
Is there only one method of testing, one test? I believe it's like a fish assay.
It's called fish assay. But today, to enter the clinical trials that are open, there is only this test. But is there the only test that will be worth to try? We don't know. We are now in the process of, first, standardizing technologies. Second, comparing technologies, right? Because in biology, nothing is black and white. All the variables in biology are continuous variables. So when we call positive and negative, we are putting a line and they're going to say, this is negative and this is positive. But it is that little gray zone. So we very likely need more than one test to clarify the gray zone.
I imagine that for the medical community at large, it's so much information, things change with such speed that right down to whether from the academic center to the private practice, all of this requires big shifts in the way physicians have treated their patients and run their practices. Exactly. Because now there's a whole new step in the care of a lung cancer patient.
Yes, you are absolutely right. And then we have to be very open-minded to all of those, because those changes are really improving the quality of the life of the patient.
And also, it's increasing the responsibility on the patient too, to be educated, to know that if treatment is ever suggested to them prior to being tested, for whatever reason, if that were to happen, it's the patient that has to say, oh, but wait a minute, I want to make sure we're doing these mutational studies on me before you decide my treatment.
Exactly. You know, the academic institutions usually are ahead of the private practice because the academic institutions are in charge of the discovery. And so usually if the patient goes to an academic institution that uses state-of-the-art technology, the patient is going to be offered. But if the patient goes to a local hospital or a private practice clinic, some of them are very aware, but they don't have the tools. They don't have these tests are not simple. We are still working in the standardization, in setting nomenclature, and setting treatment times, etc. So it's a very, it requires an updated from everybody, including the patient.
Yeah. So as with the number of biomarkers and pathways and mutations that we now have already identified, how many therapies are there available today to correspond to these molecular changes that we now know about in lung cancer?
There are about a dozen that have been open, or some few, very few, are approved treatments.
And others are clinical trials.
But the other ones are in clinical trials that we are enrolling our patients from this study, Lung Cancer Mutation Consortium. In the pipeline of the companies or in phase 1 trial, we have an enormous amount.
You've really built a case for the importance for lung cancer patients to know that they need that second opinion from an academic center. That
I will say yes.
Before they start treatment, they have got to take advantage of a place that can tell them what's happening in the area of research to be sure that they're matching their treatment to be in sync with their tumor type.
According to the molecular profile of the tumor. Exactly. I fully agree.
And we spoke about how lung cancer is this multidisciplinary disease. You are one of the components, being a research scientist, of sort of everybody working together alongside.
Mm-hmm. I classify myself as a biomarker developer.
You are doing your research at the most really exciting time that will changing the future of not just lung cancer, but all cancers.
It is, because the improvement in quality of life indeed is dramatically significant for each one of us also. Is the feedback improves the quality of life of the patient and gives us all the feedback that we need to pursuing this harder and harder.
Thank you, Dr. Marileila Varella Garcia,
Thank you.
Professor of Medical Oncology in the Department of Medicine at the University of Colorado, Anschutz Medical Campus, Aurora, Colorado.
Thank you.
You're welcome.